So protein folding is the way in which the information that's coded in the genome is decoded into life. So just about everything we think about as alive is due to proteins. There are many thousands of them encoded in the human genome. And the code is just pretty dull, actually. It's a long linear, like a long linear piece of cassette tape. And boy, you spool out a piece of cassette tape, it doesn't look particularly interesting. But if you put it in a cassette player, it does some amazing things. It's encoding this extraordinary information, beautiful music, symphonies or jazz concerts or whatever. And that's really a pretty good analogy for the human genome and for all DNA sequences on Earth that encode life. They're long linear pieces of information. And when they're read out inside of a cell, what they do is they make proteins. And when the proteins start out as long linear pieces of information too, they kind of echo that linearity of the genome. And then they start to fold. And they have to fold into very complicated shapes. You could imagine, and they have to get it just right. So you can imagine, in a sense, I'll use music again, and an analogy would be that you have a sheet of metal, and you could fold it into a trombone, or you could fold it into a tuba, or you could fold it into a flute. And if the fold, very, very different folds, very, very precise, they have to be just exactly right, or you're going to make lousy music. And that's really, I think, a pretty good analogy to the problem of protein folding, except for one additional thing, and that is that this complicated folding process, this very, very intricate folding of hundreds and hundreds of turns and twists, that has to take place in a really dynamic, crowded environment. So biology is this amazing chaos from which emerges extraordinary order. You know, you start out as a single cell, fertilized egg, and the decoding of that DNA slowly over 9 months produces a living baby, you know. And the complexity of it is amazing, but the actual environment inside an individual cell is really quite chaotic and crowded. And biology thrives on this crowding because it makes things dynamic and responsive and allows things to happen quickly. And but that crowding is very, very difficult to get these things folded properly in that environment. So that's the central problem I work on. And I guess you might imagine that it's a pretty central problem, and it plays out into all sorts of different biologies that are the focus of different aspects of the work in my lab. This crowding causes terrible problems in human biology. It can contribute to cancers. It can contribute to Parkinson's disease, Alzheimer's disease. In fact, it's really the foundational cause of those terrible diseases. When the folds don't go right, you get these clogs of proteins in the brain. But the changes in folding, and sometimes when misfolding occurs, it can create new biologies, and so that's actually another thing I work on. I work on how new types of biology evolve, how the process of evolution itself evolves. So what forces shape evolution, and one of the reasons that this becomes a central problem to protein folding biology is that environmental stress, for example, you can imagine a change in temperature, things start to heat up and chaos gets worse and worse and worse, and more proteins start to misfold, and that changes things. It changes the dynamics. It changes the way the DNA is read out into a particular type of biology. And that, however, also creates an opportunity for new biologies to emerge from that change in folding process. So I work all the way from the spectrum of trying to solve some really dastardly human diseases to trying to understand really how the foundations of evolution and life worked. Reactions will go more quickly when there's the reactants are crowded, when things are close together. Something has to interact with this and do that and give rise to this, and then that has to interact with something else. If everything is all packed together in a small little environment, it was much more efficient. So it works much better when it's working well, when it's crowded. And it's really the essence of life. Life has to be crowded like that or it really won't work. But that crowding itself creates this horrible problem of things go wrong a lot. And there are many what we call quality control systems that cells use to fix these problems in protein folding. And they're trying constantly, and they're working pretty well constantly, but they sometimes go wrong, and that can result in horrible diseases like cystic fibrosis. And that's actually due to the misfolding of just one little protein. Or in our brains as they age, the crowding problem, it's not that the crowding gets worse, it's just that the wear and tear over the years creates, as problems start to accumulate, they get worse and worse. They start having a misfolded protein here and then the misfolded one here, and they collide with each other and they form larger units and they make more and more of a mess as time goes on. So a lot of the major age-related diseases of humanity are due to problems in protein folding. So there are lots of ways in which you can imagine solving this problem. And one of the things we've sort of specialized in over the last several years has been trying to have the cells tell us what will make them better. And so we set up biological screens, for example, where we have thousands and thousands of tiny little wells of cells growing. And they're having a problem with their protein folding, and they're going to die. And then we, without prejudice, we screen every single gene in the whole genome, every piece of DNA in the whole genome for things that would make those cells feel better, things that would make those cells feel worse. And we look for chemical compounds that do that. And then we figure out how those chemical compounds connect to the genes, and we're trying to come up with therapeutic strategies that are really based on getting the biology itself to tell you what it needs. And the things that we've found actually involve lots of very specific pathways, as well as some of these general helper things. If you tune those helper processes up, it helps for a while. But there are other ways that might, if you hit the general aspects of the biology, you might perturb something else without realizing it. And so we also try to find more specific things that will make cells better.